Hi guys, this is a presentation on protein synthesis. So we looked at the central dogma the other day and we've been focusing on how DNA replicates itself. Today we're going to talk about how transcription and translation take place so we can produce an mRNA transcript and then a protein. So just a general overview of this process. We're going to look at how RNA is made from DNA and how we can use that to make the amino acids of a protein. Note that a lot of information is stored in DNA, right? This is the code that we use to produce every single protein in our body, and it's really important that we have this information stored in every single cell of our body. So just a couple of the roles of RNA. We have mRNA. mRNA is an RNA sequence of DNA that'll specify specific amino acid. So this is what's going to basically be the copy of DNA, kind of like a photocopy. tRNA or transfer RNA, these molecules are going to bring amino acids to the ribosome. Okay, they kind of are little taxis for amino acids, and they will carry those amino acids and bind correctly to the mRNA. When appropriate, they will bind to the mRNA to bring in the appropriate amino acid in the correct order. rRNA or ribosomal RNA is basically the ribosome. Okay, all ribosomes are made of a large subunit and a small subunit. So you can see on here, you have the large subunit on top, and then you have the small subunit on the bottom. We'll talk about regulatory RNA later when we talk about more control of gene expression, but that is also really important for getting a protein made. Here's just a picture to show you a size comparison. You can see the DNA is double-stranded. Our transfer RNA and our ribosomal RNA, you can see there is a lot of bonding between those nitrogenous bases. You don't really get that as much with messenger RNA, but these structures, there actually is some double stranding in certain places to give it its correct shape. This is the small ribosomal subunit, and you can just see how large a ribosome is in comparison to all of these molecules. So the first step is transcription. Transcription is going to take the DNA sequence and we're going to make an RNA transcript of it. So basically an RNA copy using RNA polymerase. Just like DNA polymerase works exactly the same way, 5' to 3', but instead of adding DNA nucleotides, we are now adding RNA nucleotides. So the big difference is it's going to be single-stranded, and it's also going to have uracil instead of thymine. The promoter is going to tell RNA polymerase where to start, and then we're going to have some transcription factors that will help the RNA polymerase bind effectively to the DNA. And this is really important. We'll spend a lot of time talking about this when we talk about regulation. So here's a quick video looking at transcription. So that was how transcription takes place. And you can see that the ribosome is really important for this process. But you also, or excuse me, I apologize. This is taking place in the nucleus. You can see that RNA polymerase is really important for this process in order to make this RNA transcript. But there also are some other things that are at play to kind of get this whole process to take place. We'll talk about regulation a little bit more, but the most important thing about transcription, okay, we're making that RNA transcript. Next step is translation. So we're taking that mRNA transcript, and now we are going to make a polypeptide sequence. Okay, this will occur at the ribosome, and let's look at how that process takes place. Okay, this is going to require tRNA, which is really, really important in order to get that correct amino acid in place. Here you can see this is an enzyme that kind of helps to actually activate and add that amino acid to tRNA. So how does all this happen? How do we know like what the correct code is? We're going to watch a quick video about the genetic code, and then we can talk about how the correct amino acid is brought in corresponding to the mRNA. Okay, so we have a triplet code, and what's really interesting about this triplet code is it's actually very redundant. You can see here by looking at this codon table down here, there are many combinations where you have a triplet or several triplets that'll code for the same thing. Looking here, we actually have 4 different ways to make lysine, and this is really nice because sometimes if there's a mutation in the DNA or a mistake, you'll still get the correct amino acid. Depending on where that mutation takes place is going to determine if it's going to alter the protein that's being produced. But we have 64 possible codons, 28 amino acids. We do have 1 start codon. Every protein starts with the same start codon, and then there are 3 stops. So this is another interesting table that I kind of thought was helpful. So you can start in the center and kind of work your way out to figure out as well. Both tables work really well, just kind of personal preference. So let's go through the stages of making a protein. The first step is initiation. So for initiation, the mRNA transcript has to come into the ribosome. Your tRNA, initiator tRNA with methionine, the start matching up to the start codon, needs to come in. Note that there's an anticodon on the tRNA, and the anticodon will have to match up appropriately with the codon on the mRNA. And then this will come into the central P slot or the P site. Once the first mRNA comes in, the top large subunit will bind, and from here, additional amino acids will come into the A site. So based on codon recognition, the next tRNA will come in, it has to match up appropriately, anticodon to codon, And then what will happen, you'll form that polypeptide bond, and this chain will keep growing, okay? And they'll shift through to the next site. And when you shift to the next site, your tRNA that is now empty will leave through the E site or exit. You'll have a new one in the P, and then the next tRNA can carry the next amino acid in. Termination, when you do get to the end, you need to hit a stop codon. The stop codon is basically a tRNA or a release factor that actually doesn't actually have an amino acid on it. So when this comes in, no bond will be formed, and that will actually release the amino acid. Okay, so this will promote a hydrolysis reaction and kind of release everything. And then the subunit parts will dissociate, and you'll have a complete protein. Okay, this is a more kind of picture of what it would actually look like on the molecular side. And then just to end for today, I have a quick video to show you how this process takes place. If you do have any questions, though, please bring them with you to class.